two speakers, each speaking for about 25 minutes, and then we'll have some time for questions after each presentation. So our first speaker is Chuan Hoi Frank, who is currently a postdoctoral researcher at the Institute for Genomic Biology at the University of Illinois Urbana-Champaign. He earned his PhD from UIUC in environmental engineering, and his research lies at the intersection of environmental engineering and synthetic biology, with a focus on engineering protein-based biomaterials and biocatalysts to address environmental and sustainability challenges, such as recovering critical resources, combating antimicrobial resistance, and converting waste into valuable products. So you can take it away.
Thank you, Dr. Wilson. Thank you so much for the kind introduction. Really appreciate the opportunity to present my work with the community. Good morning, everyone. My name is Chuan Hui. So my talk today is about how synthetic biology can be used to develop innovative environmental solutions. Here a little bit more about myself. I'm very lucky to have the opportunity to walk across different fields with different people. These fields like environmental microbiology, biogeochemistry to synthetic biology and biomanufacturing. These fields sounds a bit diverse, but they are all connected by a central goal, advancing environmental sustainability. And I'm very grateful for this experience. And of course, the people who have supported, guided, and helped me throughout this journey. Without them and many others who are not listed here, I will not be able to present today. So today my talk, I will talk about my PhD and postdoc work, which centers around two research pillars, engineering protein-based biomaterial and engineering biocatalyst. And I will also briefly introduce my future research vision. So before I dive into my research, let's take a step back. Our humanity is facing grand environmental and sustainable challenges, from climate change, resource scarcity, to water pollution and waste accumulation. So addressing these complex challenges requires curiosity, collaboration, and the development of a portfolio of innovative and sustainable technologies. And the synthetic biology is uniquely positioned to develop new tools and technologies that will advance environmental solutions. So it applies the engineering principle, design, build, test, and learn to redesign and reprogram the biological systems like enzymes, proteins, DNA, cells, organisms for useful purposes. It offers greener and more efficient solutions to some key environmental challenges, and it has its application in the environmental engineering has attracted more attention in recent years. For example, our AEESP conference this year has the first ever synthetic biology session. And also beyond the US, the UK has established a new center which explores the use of synthetic biology or engineering biology for innovative environmental solutions. So broadly, my research bridges environmental engineering and synthetic biology by designing and engineering protein-based biomaterials and biocatalysts to address resource scarcity and environmental sustainability challenges. So let me start with my first research pillar, engineering protein-based biomaterial. And I will use protein-based biomaterials for rare earth elements recovery and group separation as an example to illustrate how we design and use highly selective protein-based biomaterials for resource recovery. Rare earth elements, or REE for short, includes 15 lanthanide, scallion, and E-train. They are essential critical minerals for the green energy technology developments. In recent years, the demand for rare earth elements increased rapidly. Conventional ore mining provides a major source for rare earth elements, but the mining process is energy intensive and environmentally detrimental. In contrast, the recovering rare earth elements from waste stream provides a sustainable approach to diversifying the rare earth elements supply while reducing the environmental burden. When recycling the rare earth elements from waste stream, normally involves a process like solid waste leaching or collecting the liquid waste to obtain the rare earth elements containing wastewater, followed by the rare earth element separation and recovery, and further rare earth elements manufacturing. So as a central step, the key question is how to selectively and efficiently separate and concentrate rare earth elements from dilute and complex waste streams. Different methods like solvent extraction, membrane separation, ion exchange have been developed, but they face some challenges, including high chemical or energy inputs, generate secondary waste, and they perform ineffectively in the dilute and low-grade waste solutions due to the limited selectivity. And in recent years, protein-based bioabsorption has emerged as a promising alternative because the protein has high selectivity to the target molecule, high affinity to absorb low concentration of substrate, and requires low energy inputs due to the milder reaction conditions. And recently, due to the AI-based protein design, advances in the AI-based protein design and engineering, it's much easier to engineer the proteins. And the modulin is a naturally occurring rare earth elements binding protein that was isolated from mesotropic bacteria. And this protein is a promising candidate because its high selectivity and affinity for rare earth elements over the non-rare earth elements, which is abundant like in the waste stream. And also, this binding process is a reversible reaction, which allows us to recover the rare earth elements through the desorption. But imagine if you are using the free protein, this process will not be sustainable due to the non-reusability and the expensive single use. So to overcome these limitations, we developed a novel nanobio platform for protein immobilization, which allows us to engineer a novel bio-nanomaterial called MMP-LAN for rare earth elements recovery. So basically, we selected magnetical nanoparticle as the core to facilitate the magnetical separation, coated with the silica layer to enhance the MMP stability, and tethered the hydrophilic polymer hair for outstanding water dispersity. And unlike the traditional protein immobilization methods that face the challenge of protein leakage, non-specific protein binding, or the use of expensive catalysts for the reaction to occur, our approach uses the spy-catcher chemistry to enable stable and specific protein immobilization. So here, by introducing the spy-catcher conjugation sites on the polymer, our fusion protein LAN spy-catcher will spontaneously form the peptide bond on the nanoparticle to create this reusable and robust biomaterial. So to evaluate the performance of our biomaterial, we selected a co-fly edge nitrates, which contains only 0.03% of rare earth elements. Even in such a low REEs concentration, our bio-nanomaterial showed high selectivity for the rare earth elements, but with minimal interaction with the non-rare earth elements. As you can see here, the concentration for non-rare earth elements didn't change before and after adsorption. And after desorption, we obtain a concentrated rare earth element solution, which is about 1,000-fold purer than the initial feed. And these results demonstrate our MMP-LAN could efficiently and selectively capture and concentrate rare earth elements from low-grade waste stream, which will facilitate the downstream rare earth elements recovery and manufacturing. A key advantage of our nanobio platform is its modularity. For example, we could immobilize various fluorescence proteins on the SPI-tag functionalized magnetic nanoparticles. By changing the protein of interest, we are able to create user-defined bio-nanomaterials for diverse applications. And to expand these concepts, we harness our platform to address rare earth, intra-rare earth elements separation, which is a critical challenge due to the AEE's similar physiochemical properties and co-occurrence in waste stream. Traditional solvent extraction, as I mentioned before, has limited selectivity for rare earth elements and is also produce harmful organic solvents. So to overcome these limitations, we engineer another novel bio-nanomaterial called MMP Hanslam by immobilizing a protein called Hanslam, variants of lamodulin with a greater selectivity within the rare earth element series on the magnetic nanoparticles for intra-rare earth element separation. To evaluate its performance, we selected naturally traits, which is emerging, promising rare earth elements containing liquid waste. So we do the selective adsorption to remove most of the non-rare earth elements, and then we did a two-step desorption steps process, which contains the melanase desorption and low pH desorption. And our results show that the high rare earth elements were enriched in the first fraction, and the low rare earth elements were the light rare earth elements were enriched in the second fraction. And compared to the initial feed, the high rare earth element, the heavy rare earth elements and the light rare earth elements, the purity were increased by 64 and 195-fold. And these results demonstrate the MMP Hanslam enable enrichment of rare earth elements into light and heavy groups from low-grade natural trace, which offers a greener strategy for rare earth elements separation without using the hazardous organic solvents. So in summary, for my research pillar 1, we developed a novel nanobio platform for protein immobilization, and then using this platform, we engineer highly selective biomaterials for rare earth elements recovery and group separation. These efforts will advance the development of bio-based technologies for circular REE economy. Looking ahead, I will continue to develop the bio-based separation technologies for resource recovery, and I have two main directions that I want to explore in this area. The first direction is to harness the AI-based protein design and engineering to obtain a better metal binding proteins for more precise separation, for example, separation of the more adjacent rare earth elements. And the second direction is to program the cell as a living factories to create this self-assembly protein-based biomaterials, which will eliminate the complex protein purification and inorganic material synthesis processes, making the process more sustainable and scalable. So now I will move to my my research pillar 2, which is about engineering biocatalysts for environmental applications. So the molecular machine behind the biocatalysis is enzyme. As Dr. Francis Arnold mentioned, enzymes are catalyze all the reactions of life. They evolved to perform specific biological functions, and they make the complex materials with virtually no waste. And they are the masters of chemistry. And their extraordinary efficiency, selectivity are why biocatalysis are so powerful. So in this research pillar, I will introduce two projects. The first project is about engineering enzyme biocatalyst for organic phosphorus conversion. And the second one is is my postdoc work, which focus on engineering wholesale biocatalysts for value-added biotransformation. Okay, let's first look at the first project, engineering a renewable phytase biocatalyst for phosphorus conversion. And I will come back to this illustration later. So let's first talk about the phosphorus. Phosphorus is essential for global food production. Yes, it's a non-renewable resource. While it's essential for feeding the world, the excess phosphorus lost in the environment cause environmental problems such as eutrophication and aquatic ecosystem degradation. So there is an urgent need to recover the lost phosphorus from waste streams. In recent years, the biorefinery waste stream have emerged as a promising, yet underexplored source for phosphorus recovery. As you can see here, the core biorefinery in the US holds twice the phosphorus recovery potential compared to the water resource recovery facilities. However, more than 80% of the phosphorus in the biorefinery waste is phytates, which is a highly stable organic form of phosphorus that's resistant to the chemical hydrolysis. So as we know, the traditional take, make, use mode is unsustainable, which causes this eutrophication. To ensure the phosphorus sustainability, my vision is to convert the phytates in the waste stream, in the biorefinery waste stream, into the more recoverable and bioavailable phosphate to close the loop. And here we harness biocatalysis as a green chemistry solution for phytase conversion due to multiple advantages, including high catalytical efficiency, low energy inputs due to the milder reaction condition, and also minimal waste generation. So with this vision, I engineered a surface display phytase biocatalyst using this cell surface display technology. So in this system, the phytase, the enzyme candidate is expressed in the host cell and then automatically secreted and immobilized, localized on the cell surface to create this biocatalyst. And through this system, so it allows simple generation of the biocatalyst through simple cell cultivation, and also which eliminates the complex enzyme purification process, making the process more scalable and sustainable. And by displaying the phytase on the cell surface, we found that the enzyme showed much higher storage stability compared to the free enzyme when we store it at room temperature. And also, the stored cell can be used to regenerate a new batch of biocatalyst with a decent amount of activity compared to the biocatalyst generated from the fresh cell. And these properties supports more flexible deployments of pre-cultivated biocatalyst and supports the on-demand generation of active biocatalyst for the phytase conversion application. However, one potential limitation is that this phytase can be inhibited by the substances in the biorefinery waste stream. So to kind of address this limitation, we used a deep learning-based approach. We use a muse compute, a structure-based neural network to identify the stabilizing mutation. And our results show that multiple variants of the phytase showed significantly higher activity than the wild type. So now I will briefly talk about the second project. So where I engineer a wholesale biocatalyst for waste-to-value biotransformation. So in Illinois, an agriculture-intensive state, the cellulosic biomass and agriculture residues are low-cost and abundance feedstock. So my postdoc work is about engineering non-conventional yeast called lodotolular tolulyse to convert the cellulosic sugars in the biomass into a platform chemical called triacetic acid lactone to achieve this value-added biotransformation. And as an effort to achieve this goal, I am developing the first automated high-throughput screening pipeline, which combines the high-throughput random mutagenesis and high-throughput colony screening. Use the robotic in the biofungi to achieve this efficient value-added biotransformation. So overall, for my research pillar 2, the renewable phytase biocatalyst work opens a new avenue for circular economy, and my postdoc work will develop a new platform to accelerate non-conventional yeast engineering for sustainable value-added biotransformation. So building on these foundations, my future, I will continue to develop an enzyme and microbial biocatalyst platform for waste-to-value biotransformation. And I believe by converting the renewable and organic waste into high-value bioproducts, the biocatalysis offers a greener alternative to the petroleum-based synthesis, which will not only address the grand environmental challenges, but also contributes to the circular and resilient economy. So after my graduation, I have thinking, can I use the biocatalysis to address other critical challenges? And one such challenge that's caught my attention is this antimicrobial resistance. So antimicrobial resistance occurs when the antimicrobial agents are ineffective to kill the microbes, and it's a top 10 global public health threat, causing 1.27 million deaths annually. So there is an urgent need to mitigate this growing crisis. And more studies shows that the wastewater is a hotspot for the antimicrobial resistant bacterial and antimicrobial resistant genes. Even after the bacterial die, its DNA, extracellular ARG, can still stay in the environment for a long time, up to years. And this cell-free ARG can confirm the consistency through this horizontal gene transfer mechanism, including transformation, conjugation, and transduction. So, and conventional wastewater treatment processes such as UV or advanced oxidation processes are not effective to remove this ARG. So my vision is to harness the biocatalysis as a green chemistry solution for the EARG degradation, which will limits the horizontal gene transfer, thereby reducing the resistance spreading. So with this vision, and also to see the grants, we engineered a nucleus biocatalyst for AMR mitigation in the wastewater. So we engineer a biocatalyst called SDN. And in our test, we found that this SDN could very efficiently degrade a plasmid carrying ampicillin resistance marker in the secondary afferents, while the blank control and negative control didn't show the degradation. And to further confirm the degradation, we transformed the treated plasmid into the E. coli competent cell. And as you can see here, for our biocatalyst treatments sample, there is no transformation, but for the blank control and negative control, it shows a high transformation efficiency. So these projects will provide a new biocatalytical strategies for AMR mitigation by targeting and degrading the extracellular ARG in the wastewater. So overall, my research is my research vision is to advance environmental sustainability and a circular economy through the power of synthetic biology by applying the tools from molecular and synthetic biology while integrated with engineering principles and emerging AI approach. My research will focus on engineering proteins and microbial systems to address pressing environmental challenges in resource scarcity, public health, and waste valorization. And more specifically, I will design and engineer protein-based biomaterials, enzyme biocatalysts, and microbial biocatalysts to deliver new solutions to critical environmental challenges. Yeah, thank you very much for your time. Sorry, I cannot see your face while I'm presenting. Yeah, I'm very happy to take any questions. Thank you very much for the opportunity. Thank you.
Yeah, thank you so much. Great talk. So we have some time for some questions. So if you want to ask a question, raise your hand or just unmute.
I have a question, but I couldn't find the hand.
Go ahead, Liz.
So my question is, can you say a little bit about what the characteristics of the enzyme landmodulin and some of the other enzymes that you were using that makes them so selective for the rare earth elements? What functional groups do they have?
Yes, sure. That's a very good question. Let me find if I can find some slides. Yeah, definitely. It's has like those enzymes, those proteins have their specific structure that allows them to have high coordination with the rare earth elements. And those amino acid residue are those like glutamate, aspartame, which has this carboxylate-rich group, which will provide the strong coordination with the rare earth elements. Let me quickly go to find the slide. If you want to see here. So yeah, so here you can see D-aspartate, another aspartate. So yeah, this provides strong coordination, and also this binding process between the lemodulin and the rare earth elements is a cooperative process. That means when you bind one rare earth element and the effect is cooperative, it will bind more, which makes it more stronger. And also for this LNM protein, it has a different strategy, which kind of rely on this dimerization. So when you bind the light rare earth elements, it will form this dimer, and it has this carboxylate shapes and hydrogen bonding network to coordinate like the coordination bonds between the light or heavy rare earth elements to the protein.
Okay. Thank you.
Thank you very much for your question. Yeah. That's a very good question.
Go ahead, Lee.
Yeah, so thanks for the nice presentation. Really enjoyed it. You packed a lot of material in there, and I learned a lot, and so that was awesome. I also wanted to ask about the rare earth element part, and so just to remind me, I came in a few minutes late. Those were nanomaterials that you're using, is that correct?
Yes, I'm using a magnetical nanoparticle functionalized with the highly selective protein.
Okay, and is it on some kind of support, or it's actually a nanoparticle?
So the nanoparticle has multiple parts. So the core is the magnetical nanoparticle, and then on the outs layer is a silica layer, and we also like incorporate polymer, hydrophilic polymer to increase the water dispersity of the MMP. So it's kind of I think it's a hybrid material. Yeah.
Yeah, but you were running all of those in batch mode, is that right then?
Yeah, exactly. That's also kind of related to our future vision that I'm thinking about more like a self-assembled biomaterial, which will allow like continuous or semi-continuous rare earth element separation. Because for the nanomaterial, it's not easy to transfer from the batch mode to the semi-continuous mode. Yeah.
Well, I was actually thinking, if you're able to put it on a support or something else that you can run in column mode, not necessarily the absorption side, but now you can have kind of an elution gradient, and rather than just separating
Yes.
like the light and heavy rare earths, like maybe you can actually separate fractions of individual
Exactly.
compounds and remove the need for downstream like separation processes that are very intensive.
Yeah, exactly. And some people already put the enzyme, put the protein on like agarose. The agarose can be put on the column, and then they do some more fine-tuned separation of rare earth elements. Yes, exactly.
Okay, cool. I look forward to seeing how it progresses. Congrats.
Thank you. Thank you. Appreciate that. Yeah.
And then just one more quick question from Mahmood.
Hi.
Hi, Dr. A. It's a great presentation. Thank you so much. So I have a couple of questions. I just want to know, like you have mentioned that for RE binding those kind of amino acid sequences and also those carboxylate group and also the EF hands geometry, provides a great, has a great role in those kind of RE binding. But I'm just wondering, like, how can we utilize those kind of leverages to separate, REs from non-RE? How can we use, how can we think about like those kind of EF hands or carboxylate group or amino acid sequences? Except those things, how can we separate REs from non-REs? Because in the real wastewater, those kind of mines or, mining wastewater or some other kind of wastewater, those have like a lot of base metals and REs. How can we selectively isolate them?
Yeah, that's exactly why we want to select a highly selective protein like lamodulin, because it has those like key amino acid residue which show high coordination with the rare earth elements, but it shows minimal interaction with the non-rare earth elements. So that's kind of amazing about the biology, I think. So the mesotropic bacteria has evolved such an enzyme as a cofactor when it's used this ethanol dehydrogenase for this, like use the rare earth elements as a cofactor. So the microorganism itself already evolved such a mechanism to selectively capture the rare earth elements from the environment and then use them as a cofactor for the key enzyme, methanol dehydrogenase. Yes. So I'm not sure whether that's answer your question. I think maybe due to time, maybe we you can send me an email or I can follow up. Yeah.
Okay. Thank you so much.
Yeah. Thank you so much for your question. Yeah, I appreciate it.
Well, shall we go to the next speaker, Hunter Kwan? Are you here? And if so, can you share your screen? I saw you join the meeting.
Hear me?
Yes.
I'm turning everything on.
Okay.
Sorry about that. The camera should be on.
Okay.
You see
And
me? Good.
so you have your presentation that you could share? Great. Okay, so let me, and we will stick to our schedule. So I'll introduce you briefly. Hunter Kwan, our next speaker, is a postdoc at Arizona State, and he earned his PhD from UC Irvine. And his research focuses on quantitative microbial risk assessment, environmental microbiology, and computational modeling for risk assessment and water and wastewater treatment. Okay, so whenever you're ready, go ahead.
Okay, thank you very much. I switched to presentation mode, so hopefully you can still see my correct screen. Thank you very much for inviting me today to present a little about me and my work. My title of my presentation today is Integrating Data, Microbes, and Society, Modeling for Sustainable Water and Health. As mentioned, a little bit about my background. My academic background is entirely in civil and environmental engineering. with my bachelor's at Oregon State University, my master's and PhD in civil and environmental engineering at UC Irvine under Dr. Sunny Jiang. And for the past about 2 years now, I have been at Arizona State University with Dr. Carrie Hamilton in the Biodesign Institute and their School of Sustainability and the Built Environment. Just to give a little outline, I'm going to break up my research and my experiences into sort of 3 main topics or areas and talk about some past and ongoing work in each one, and sort of sandwich that in between some overall intro and motivation and future directions of my work. Now I've got some nice little pretty photos here, all related to water, just to say that obviously water is everywhere and we use it for a number of different things. And water that may seem ordinary or clean, we use it in so many different ways, and depending on its source and its use, it can still definitely carry some health risks. So my research focuses on trying to better understand, predict, and prevent these risks from things like pathogens, like waterborne pathogens such as Legionella and Haemophila, all the way to things such as antibiotic resistance, and trying to develop quantitative models to measure these contaminants and these risks, compare them against each other, and ultimately support better decision making for public health and sustainability now and moving forward. And so that's my overall sort of purpose and direction. And I'm ultimately motivated by a number of things, namely water shortages and sustainability. This big, flashy news article saying that 5 billion people will face water shortages by 2050. That right there, I think, speaks for itself. And then some more health-related ones that I've already mentioned are implications for rising concerns around waterborne diseases, such as Legionnaire's disease, which is shown in this graphic on the left side, which has been rising and continues to rise all around the globe, such as in the US and in Switzerland for the past couple of decades, and then for antimicrobial resistance in the environment, which is considered an emerging threat and is also predicted to surpass cancer in terms of mortality by 2050. And then also the need for improved education, community engagement, and overall equity in both access and use of clean water all around the globe. So moving into my first main topic or research area, quantifying hazards in the built environment. I'm going to talk a lot about waterborne pathogens because that's been sort of the backbone of my work in the past. And waterborne pathogens in building water systems, or premise plumbing systems as they're sometimes called, are an ongoing concern, such as causing respiratory illnesses. You might have heard of Legionnaire's disease, and over $2 billion in annual healthcare costs just in the US. And my guiding research question around this is, how do we quantify and then ultimately try to prevent these risks around these waterborne pathogens? A really big part of my research since doing my PhD and even now is the quantitative microbial risk assessment framework. And for those of you who are maybe less familiar with it, it is a framework that's widely used to assess the risk of illness, infection, or even death, some sort of adverse outcome from exposure to a given pathogen. And it's entirely quantitative, as the name suggests, and it's based on data and some modeling techniques such as Monte Carlo simulation to capture uncertainty and statistics and the best available data in applying this framework. And it's described and shown here on the right side. And I'm going to sort of really quickly walk through an example of how QMRA works with a study that I did based on water in the US Virgin Islands after some major hurricanes. So I've never been there myself, unfortunately, but this picture makes me want to visit. The US Virgin Islands are a tropical destination in the Caribbean, and where the samples were collected were part of the United States territory. And interestingly enough, in the Virgin Islands, there's actually a law that requires that most buildings be constructed with a self-sustaining potable water system, such as a well or a rainwater collection system. So rainwater collection cisterns are commonplace in the Virgin Islands. You may recall back in 2017, there were back-to-back hurricanes in the region, Hurricanes Irma and Maria. These pictures are from the Virgin Islands. So these hurricanes were catastrophic, lots of destruction and impacts to the water quality all around. And then some further graphics here showing these rainwater collection systems that I talked about, rainwater catchment on the right, and a sort of barrel or cistern on the left. So ultimately, the roofs are designed to collect rainwater and then have like a gutter sort of piping system to store it in a cistern or barrel. And then that would be really the primary water source for a lot of households and buildings. So just to really kind of throw at you an example of how QMRA works, maybe in this case, we went in and we want to look at what might the risk be to a given pathogen. In this case, over 80% of water samples collected from cisterns after the hurricanes tested positive for Legionella and Haemophila. So that sets sort of the foundation and motivation to do a QMRA on, well, is the amount of Legionella in these cisterns, does it pose a risk? Does it pose a health threat? So we identify the hazard, which is Legionella in the untreated cistern water, try to best characterize it and measure it in some way, and then move to our next step in the framework, exposure assessment, which is really creating a quantitative model to best estimate the dose of this pathogen that a person might be exposed to for a given scenario. And the chosen scenario here is daily showering, because this is something that pretty much everybody does, as they indicated by surveys that were collected at the time, and they use their rainwater to take showers. And not only that, but showers create aerosols, which is one of the primary exposure routes of Legionella and Haemophila to get into your lungs. So really just to say that through QMRA, we apply a bunch of equations to best estimate the dose, the quantitative dose response, which is how much of that dose may then go on to cause an adverse outcome like an infection, and then characterize this risk based on any number of exposures, whether you take a single shower or whether you're showering throughout the year. And then use, like I said, some statistical techniques like Monte Carlo simulation to sort of create some uncertainty bounds around this. So that's just really how QMRA works, and I've used it a number of times in recent studies where I, through Legionella in building water systems, tried to assess and lead some studies. So I mentioned this one where this example comes from. Ultimately, the main conclusion of that study was based on post-hurricane samples and surveys. The annual infection risk was above a 10 to the minus 4 threshold for the detections, and that is shown in these results and is published in the journal Water. I've also worked on more recently studies of doing a meta-analysis of looking more into how do we actually intervene and treat Legionella once it's in a water system. So there was a systematic literature review of the reductions achieved through applying interventions at different scales and different types of buildings, all the way from lab scale up to residential and commercial buildings, and also in healthcare facilities. This is some of the results of that. Just to say that in general, we see positive log reductions in most cases for Legionella, but for non-Legionella contaminants, the studies that happen to measure other things like metals, copper, lead, or even disinfection byproducts, whoops, that circle was supposed to appear, we see negative log reductions, or in other words, the measures of these contaminants after treatment were higher than they were before. So that was one of the main findings of this study and provided some motivation for future work. In addition, I talked a lot about inhalation of these aerosols, but aspiration, in other words, when water or aerosols go down the wrong pipe while you're coughing or drinking water, they've been talked about quite a bit in the literature as a potential route of Legionella transmission, but we went ahead and applied models to try to see how it actually looks when we quantitatively compare it with inhalation. And through a number of different simulations, we found that the risk was indeed higher than for inhalation exposure, and therefore concluded that it's an understudied route in terms of exposure science and assessment. Some ongoing work I have in this area is, like I said, with the applying interventions, having different contaminants increase or decrease after the intervention, I want to assess the trade-offs of these different contaminants. That is, back in terms of risk and exposure, if we go in and we treat a system, we apply, let's say, a chemical treatment or a physical treatment such as flushing or increased temperature, are we really going to see success or an intended outcome? That is, maybe we reduce Legionella, we successfully reduce it by a few logs, but maybe in that same system, depending on the water source and how it's being used and sort of the makeup of the infrastructure, we ultimately see increases in things like lead or in disinfection byproducts. So this was a recently ongoing work. It's currently under review, so hopefully I'll have more to share about that coming soon. And ultimately trying to link models with reality. So not just in predicting the risk, but in actually comparing infection risk with epidemiology. So some ongoing work that I have going on with the New York State Department of Health is using a very large data set of Legionella detections and prevalence estimates in healthcare facilities throughout New York State, and sort of linking them back to a risk assessment framework to really compare at the end of the day, does modeled risk actually compare with observed risk or actual outcomes such as outbreaks? Okay, moving ahead to my next sort of topic area of my research are the environmental dimensions of antimicrobial resistance. So this little graphic here is just to give you a little example of how antimicrobial resistance works if you're not as familiar with it. Some bacteria out there are genetically resistant to an antibiotic. So when you take an antibiotic or when they're exposed to an antibiotic, they will survive. Ultimately, the antibiotic will not kill them. So maybe the antibiotic kills the rest of them in the population, but a few of them that are resistant will survive. Those surviving bacteria will then grow and spread, and that is how antimicrobial resistance spreads in different environments, such as in the body or in wastewater. I mentioned earlier that it is also predicted to be not only a current issue, but an emerging issue in the next couple of decades. And this graphic here shows that it's predicted to get as high as 2 million deaths per year just to resistant bacteria by 2050. And these numbers are also predicted to exceed cancer or in the same time frame in terms of mortality. And my guiding question here is, what role do environmental pathways play in antimicrobial resistance dissemination dynamics, and then again, public health risks? Some of my recent work in this area is again using the QMRA framework, but sort of adapting it and trying to account for antimicrobial resistance and antibiotic resistant bacteria pathogens. So I applied recently a new dose response model to account for resistant bacteria in the context of wastewater reuse and reclaimed wastewater being used for the irrigation of food crops. So what I'm showing here is risk outcomes, both as a quantitative probabilistic risk estimate of annual risk of infection, plotted as against the increase of antibiotic concentration. So without maybe getting too deep in the weeds here, what this model does is it accounts for how much of the bacteria or how much of the dose that you might be exposed to in a given scenario, how much of those are resistant to an antibiotic, and if those bacteria were then exposed to antibiotics, which ones are likely to, how many of them are likely to survive and cause an infection. And this was based on real data collected from greenhouses in Spain from a collaborator. And I didn't mention this on the last graphic, but bacteria can pass their genes and their resistance genes to each other through different mechanisms, such as conjugation and horizontal gene transfer. And this is also a highly uncertain area in terms of quantitative modeling. And some recent work was really trying to take a step towards trying to include this in a more dynamic model. And the first step there was, well, maybe we should do a literature review and see in different environmental contexts, what does that conjugation actually look like? What are the numbers? Are they higher or lower in different contexts or for different pathogens, for example? And maybe it's not ultimately too surprising, but we found that the variability in this was gigantic. There was a 12 log range across conjugation rates once they were all normalized across the same units, depending on the source and the conditions of the experiment and the bacteria and the source of the water itself. Oh yeah, shown here. Moving forward with that literature review, I have some ongoing work related to trying to actually account for this conjugation in a more dynamic model where we have some results, some recent results, they're still kind of happening for single donor, single recipient experiments to better characterize conjugation and model parameters. So again, linking this back to QMRA and how can we actually model this dose and how these bacteria may or may not dynamically change between, say, being applied to an irrigation setting and a human consumer. Which again comes back to here where we have some measurements of detection of both resistant and susceptible bacteria in agricultural settings, and trying to look at the outcomes based on the agricultural locations, the different water sources, that is whether the water being used for irrigation is coming from a municipal source or an open water source such as a pond or a canal, and how it's being applied. So these are just some preliminary results where on the left side, panel A, pretty much all of the samples were below the detection limit for resistant bacteria, except for one location where we had some positive results. And once we translate these results of the cefotaxime resistant E. coli into a risk framework, we see elevated risk for that single location compared to the rest of them. And then in my final research area, the social dimensions of water safety and sustainability. This is something that ever since that Virgin Islands study where we had a survey that was conducted for the residents to understand their water uses and how they understood the risk that may have come with it, especially at the time after the major hurricanes. It's something that's I've been really, really interested in, and more so lately, I've luckily been able to get more involved in. And that is because vulnerable communities face disproportionate water risks without effective engagement and communication. So my guiding question here is, how can we use community engagement and communication to improve water safety and sustainability? So I talked about that with the Virgin Islands, where I analyzed the results of the survey, where we asked them questions like, what do you use your water for? Where is your water coming from? Are you buying bottled water? Are you drinking your rainwater, cistern collected water? Are you using it for showering, for food washing, for things like that? And then questions like, how safe do you think your water is? Safe, somewhat safe, or unsafe? And at the time, because of the hurricanes, the government had sent out an water boiling advisory, which was to say to boil your tap water before drinking it or using it for consumption. So we asked them, have you heard the advisory from the government? Do you think the government has done enough to let you know about the safety of your water? And do you think the government has done enough to provide you with a safe water supply? One of the main findings that I showed here in the different color scheme was that there was more of a trending towards statistical significance between high and low income individuals and how they viewed the safety of their water. And then more recently in the state of Arizona, specifically in the Phoenix area, we implemented another survey to better understand public acceptance of advanced purified water, asking them questions like, are they familiar with advanced purified water, where it's coming from, how it's being treated, and how safe it is, to understand their perception of water risk and water safety. And asking them questions like shown here in the graph, what they would be willing to use it for if it became a new source of water. And really through these surveys and open dialogues with the consumers, we better identified some of the key barriers and strategies really to build trust. And I think that's what this all comes back to, is using tools like these, using increased community engagement, surveys, and communication to both build trust with the community and increase their awareness of general trends around sustainability and how we can improve our water use and our water supply. A little bit of ongoing work with this is I'm working with the Center for Produce Safety, specifically on irrigation practices, and to get grower feedback to create, I'm calling it a stakeholder-focused risk tool. This is already an older graphic, but really just an app that farmers and growers can really simply go in and use and try to describe or quantitatively put in and describe where their water is coming from and what types of foods they're irrigating with, and spit out a graphic and a recommendation related to risk. So if they were to detect indicator bacteria or pathogens in their water supply, we would use this app to really simply give them some options about what they can do to prevent risk and to mitigate it. And a similar idea around biosolids. So just the general use of and acceptance of biosolids from both consumers and growers. And this is in the very early stages, but implementing both community and industry surveys just to get an idea of the understanding and perception of the use of biosolids in the state of Arizona. And I kind of want to just throw in a little bit of my teaching and mentorship, and then some of my future directions. My teaching experience, I've been a TA and a guest lecturer a number of times, but just this last year at ASU for the Civil and Environmental Engineering Department, I was the instructor of record for a data science course called Data Science for Civil and Environmental Engineers, which is now required in our department and our program. And I had a really great experience doing that. And I've mentored a number of students in research from high school all the way through graduate level in different quantitative research projects, teaching about how to do QMRA, what is it, and how do we sort of start from the ground up on a QMRA focused research project. I've worked with ASU's SEEN program, which is the Science and Engineering Experience Mentor Program. And at UCI, I was a mentor and tutor in the Office of Outreach, Access, and Inclusion. And very recently, I'm really excited about this, I've actually become a Phoenix Water Wrangler, which means I'm a volunteer where I get to go hang out at booths at lots of different types of events and spread the word and talk to people about water conservation and sustainability. And I'm especially excited about these 3 stickers that they gave me, which are sort of their mascots for potable water, wastewater, and stormwater. Some of my future research plans, my last little bit thing here I want to mention is for each of my research areas. I talked a lot about Legionella and QMRA, but some of the more recent interventions and trade-off work is where we've begun to identify that there's holistic challenges going on. So I want to look more, like I said, holistically at how things like climate change, infrastructure, water management in buildings, and surveillance, they all can interact with microbial risks and these other contaminants, not just Legionella or not just E. coli, for example. And to look at sort of the bigger picture and how these real world hazards could pose threats to specific communities, like in healthcare facilities. I want to continue to better quantify and characterize parameters like around AMR, like conjugation and rates that may really drive how these populations behave in different contexts and different environments. And then again, tie it back to public health risks. And then lastly, at sort of my interface of water safety and society, I want to continue to investigate how people perceive water safety, water use, and how different communities and different locations all around the world, locally and abroad, may accept or reject different sustainability practices, and why. Why would they? Why is their first maybe instinct to accept or reject them? And I really think that this varies significantly by context and by location. So that's something I want to look into as well, not just at home, but abroad, like in low and middle income countries. All right. Thank you very much for your time. I think that's
Thank you.
good on
Yeah,
time,
so
yeah.
we're right at the time.
Okay, great.
I think that we're right at noon, but you started a little late because the last speaker, we got behind. So we have time for just one or two quick questions, if that's okay.
Yeah, sure.
So go ahead with your questions, participants. Lee Blaney has raised a hand, so please go ahead. Oh,
All right. Thanks, Hunter, for the presentation. I enjoyed it. I was a little bit curious about your antimicrobial resistance work, and maybe this question is like a little bit further afield, than what you presented. I think you kind of focused on wastewater reuse and kind of thinking about that as being a driver. I wanted to ask you a little bit more about like agriculture. I think, my numbers might be outdated, but at least as of a few years ago, we use like 70% of antibiotics in agriculture. And so I'm wondering, have you thought about like applying any of your tools to agricultural settings or agricultural communities and looking at that, what that means, for antimicrobial resistance development in people living in those communities versus maybe more urban areas that are, seeing water reuse?
Yeah, good question. It's interesting because most of my work in this has been in agricultural contexts for food crops, but it so far has been, very, very, I'm going to say study specific, but you're right in that in these contexts with animals and with food, antibiotics are being used. So something that is part of my earlier work, very briefly, but it's something I want to explore in the future, is how do the antibiotics themselves drive these models? So looking at it more of a pharmacodynamic, pharmacokinetic version of the model. So that is absolutely something that I'm curious about next and want to sort of look into next. But the ongoing work doesn't sort of measure that or account for that. We're just looking at sort of population makeup. How many within the sample are carrying the gene that is resistant to cefotaxime, for example.
Got it.
So I think it definitely can drive, once we sort of better characterize how these bacteria populations may change post exposure to an antibiotic or once applied to a crop, and then we can model the populations after that, between human exposure.
Yeah, thanks so much.
so thank you, Dr. Kwan. I don't see any more questions. So Jessica, do you want to say anything else?
Yeah, I just want to thank both of our speakers
Yes.
again and all of the attendees. And to remind everyone that the recording of this seminar will be posted to the AESP YouTube channel, and that we have our third seminar a week from today on Friday the 21st. So we hope you're all able to attend that. And thank you, and enjoy the rest of your Friday.
Thank you.
Thank you.
Thank you.